Hello, my name is Sebastian Köhler. I'm a third-year PhD student in the SyNaBi group here at TIMC and the large-scale structures group at the Institut Laue Langevin. I'm supervised by Donald Martin and Marco Maccarini here from the SyNaBi group and Giovanna Fragneto at the ILL. My PhD project's topic is the structure-function relationship of biomimetic membranes with incorporated membrane proteins, and as the title indicates, I'm interested in how the activity of a membrane transport protein is connected to the structure of the surrounding lipid bilayer. The motivation for this is twofold. From a basic fundamental biophysics point of view, we want to study interactions in the protein membrane system to see how its structure affects incorporation and under which conditions a stable protein membrane system is created. On the other hand, it's connected to a very applied project, the development of a biomimetic energy device, which makes use of membrane transport processes to create power for example, biomedical implants. The concept is inspired by biological processes. A human does not need batteries but runs on food, the classical fuel cell. On the cellular level, part of this is achieved by the exchange of electrolytes with the surrounding. This exchange is carried out by membrane proteins in the cell wall. We want to use this kind of ion exchange across the membrane to create a transmembrane potential, which again can be transformed into an electric potential by placing electrodes on either side of the membrane. At the heart of such a device would be a planar lipid bilayer separating two compartments. Incorporated into this membrane are antiporter proteins, which move sodium in one direction and protons in the other. The protein we decided to investigate is the sodium proton antiporter A, in short NHAA, which transports two protons for one sodium ion across the membrane. This way, a net current across the membrane is created as more positive charge is moved in one direction than the other. For such a device to deliver optimal output, the protein's transport activity must be optimized. This can be achieved by increasing the protein's activity or by incorporating large amounts of protein into the membrane. We are interested in which structural factors of the lipid membrane environment affect these two parameters. The model membrane system which we are using are so-called tethered lipid bilayers. They consist of a planar lipid bilayer which is chemically connected to a surface by tether molecules. This leads to different advantages. Tethered bilayers are stable for up to several months, and because of a hydrophilic part in the tether molecule, a water-filled physical separation from the solid support is created. This prevents, for example, protein surface interactions. In my project, I investigate three different kinds of tethered lipid bilayers, which differ in, for example, the water content in the submembrane space or the rigidity of the connection between membrane and surface. With these systems, I characterize the incorporation of NHAA. For this, I mainly rely on two different methods: neutron reflectometry and electrochemical impedance spectroscopy. Using impedance spectroscopy, I study the electrophysiological properties of the membrane, including protein activity. With neutron reflectometry, I characterize the nanostructure of the protein membrane system. In neutron reflectometry, the intensity of a neutron beam reflected from a planar surface is detected for different angles. By finding an appropriate model, the average molecular composition perpendicular to this surface can be determined with sub-nanometer resolution. I performed reflectometry experiments on the three different tether systems. The main work went into a polyethylene glycol tether system connected to a gold surface. With this system, we are able to produce highly covering membranes and incorporated large fractions of around 40% protein without destroying the bilayer structure. For electrophysiological characterization, I used impedance spectroscopy. With this method, a system's response to an external alternating current signal of varying frequency is measured. By finding an equivalent electrical model circuit, parameters like a membrane resistance can be obtained, which describe the amount of defects in a membrane or the activity of a protein. Using this method, I refined the lipid composition for low defect membranes, which is necessary for the creation of the transmembrane potential. At the moment, I am working on the characterization of protein activity by impedance spectroscopy. As the protein creates a transmembrane current, this influences the resistance of the lipid bilayer, which is again visible in impedance spectroscopy. Combining these two methods, I identified favorable properties for the biomimetic fuel cell and was able to produce stable membranes with high fractions of incorporated membrane protein. With the analysis of the neutron reflectometry data from the other tethering systems, I will be able to evaluate their suitability for protein incorporation and their influence on its activity.